In this video, I will be presenting an Excel interface that I built to help biologists analyze flow cytometry data. The interface provides a user-friendly bridge between raw flow cytometry data and TASB Flow Analytics, or TASB for short, which is a flow cytometry analysis package in MATLAB. I designed all of the features in this project. Flow cytometry is a standard tool used for the rapid multiparametric analysis of individual cells within heterogeneous populations. It captures and measures the intensity of light emitted from each cell suspended in a stream of fluid. A flow cytometer consists of the fluidics, optical, and electronic systems. The fluidic system uses sheath fluid to hydrodynamically focus a cell suspension through a small nozzle, forming a single file line of cells. As each cell passes through a laser beam, scattered light is detected. Fluorescence detectors measure the fluorescence emitted from stained cells, which can be used to separate cells based on protein expression. From there, the detected light travels through filters and mirrors, going to the correct wavelength detector, and is recorded by converting the light intensity into a voltage pulse. Though flow cytometry is a powerful biological assay that can quantify fluorescence precisely for large populations of cells, interpreting flow cytometry data requires a complex series of calibration and analysis steps, in which many biologists do not have expertise. One challenge involves spectral overlap, which occurs when two or more fluorescent stains have overlapping emission wavelengths that could be measured by a single filter. For example, in the left figure, FITC and PE can both emit light with wavelengths between 555 and 600, which the PE filter can detect. This means that some cells with FITC could accidentally be detected with the PE filter, resulting in incorrect classifications. Without thoroughly calibrating the fluorescent stains, it is very difficult to account for spectral overlap. Another challenge of flow cytometry is the process of identifying different populations of cells using gating techniques. Currently, most gating is done by hand, which introduces the potential for subjectivity and human-induced error. Furthermore, gating is typically done on axes with uncomparable units of fluorescence, making it impossible to compare results from one experiment to another. The flow cytometry analysis package, TASB, attempts to address these challenges. The typical workflow begins with the creation of a color model, which calibrates and translates raw flow cytometry data into comparable unit data. The color model can then be used for additional analyses, including the batch processing of experimental data, comparative analysis, and transfer curve analysis. I created an Excel interface which acts as a user-friendly and unified platform for current TASB data analysis tools. Biologists without a lot of prior coding knowledge can set up and customize analysis workflows in Excel instead of MATLAB. The interface consists of three major components: a spreadsheet template, a MATLAB backend, and Visual Basic functions connecting the two. The template is organized as a series of sheets including an experiment overview sheet and a variety of analysis sheets. The template is flexible, allowing users to run multiple analyses at a time. Visual Basic is employed to implement run buttons that call the MATLAB backend to execute and extract session logs while analysis is running. All of these features work together to form an approachable interface, which integrates all of TASB's best features into one cohesive platform. Taking a closer look at the MATLAB backend, when a user clicks on a Run button, a Windows COM interface opens and runs the TASB Analyze from Excel function. First, a template extraction object is created, which is used to obtain information from the Excel template. From there, the correct analysis functions are called. To get a better idea of how the Excel interface works, I will walk through a few sheets of the template. The experiment sheet contains all of the overview information, including the different conditions that were varied in an experiment, and file name templates that are used to generate file names in the sample sheet. The sample sheet includes a list of experimental samples with their file names and condition information. This sheet is also used to run batch analysis. The calibration sheet has all the information needed to create a color model. When the Run button is pressed, VBA functions connect to the MATLAB backend. One of the main features of the Excel interface is the updating session log. The session log displays all notifications, warnings, and errors from TASB. Users can also continue to edit the interface while analyses are running in the background. Both the session log and user editing features work because of cooperative multitasking, which involves creating a copy of the interface from which the MATLAB functions would be called, and then linking everything back together when the session log is updated or after an analysis completes. Once the color model is generated, the selected figures are automatically copied into the calibration figures sheet. There are additional sheets for comparative and transfer curve analysis. More analysis settings can be edited in the optional settings sheet. The Excel interface allows more researchers to design complex multi-stage workflows to analyze their flow cytometry data. It is currently being used by several synthetic biology research groups. Based on their feedback, I constantly strive to make improvements to the interface to create a more unified and smooth user experience. For more information, please visit the TASB GitHub page.